Hi, my name is Alexa Alvarez, and today's date is July 28th, 2020, and we are working on Lab 7, which is bacterial identification through functional media. And the exercise we are working on is motility testing. So this is my card. Okay, so we have I was in the process of straightening out some paper clips, so these are gonna sit in alcohol to sterilize. We are going to use our E. coli and epidermidis cultures, and we are gonna pretty much check for motility by using these agar tubes. We don't have to melt these or anything. We just have to, we're gonna stab, place some of the cultures in each one and let them sit for a couple days, hopefully not any longer. And we are gonna see if we see any motility within the media or the agar. So right now I'm in the process of straightening these paper clips, and we're gonna put some alcohol. Hopefully this doesn't fall. In alcohol. So those are gonna sit there for a little bit. And now we have to label some of our test tubes. Put you on this side. We're gonna label some of our test tubes with permanent marker, and for the agar. So one of them is gonna be E. coli. So we're gonna label this. And then the next one, we're gonna do epidermidis. And we have to light the candle. We're gonna disinfect the test tubes that have the motility media in there, and then we're going to let the paper clips dry and then work on transferring some of the bacteria over. Actually, I think I have to correct this. We're not using these. We are using actual live cultures. So these guys. Yes, so we're using these. This away. Okay, so I had originally taped my cultures just because I didn't want them to open up where I was storing them in the fridge. So I had to cut the tape. So now we are going to start transferring. So we have to open these, and with our candle we're going to sterilize the opening of the tube a little bit. We have to shake this to let it dry. this off. Looks pretty dry. I'm gonna open this. We have to collect some cultures. Now we're gonna stab this, trying to be really careful not to touch the edges. Okay. And then we have to sterilize this again, so I don't know if you can see, probably be hard to tell, but you can kind of see the line. It probably goes down to like here of where we stabbed the medium. Close this. I'm gonna close this. Now we're gonna do the same thing with the epidermidis. Let's take this out of here. Let this air dry. And again, we have to sanitize or sterilize the opening. So again, this has to air dry. Okay, now we're going to collect some cultures. This one looks a little weird. Again, we're going to stab the agar. And again, you can kind of see, well, you can see it a little bit. You can see where the line is. Just this one though, I kind of went in at an angle. Hopefully it still works though. Okay. And then we're going to sterilize this again before we seal it. And now I'm going to close that. So now I have to take some pictures, upload them, and then these have to incubate for about 48 hours. If it's not ready within 48 hours or I don't see any movement, I will leave them for another 24 hours. So today is Tuesday, so I'm hoping we have some growth by Wednesday, Thursday night, and we'll see how it looks. So I think I'll see you in a little bit. Hello. So today we are continuing our part 2 of our experiment. Oops, sorry. There we go. So we're continuing the second part of our experiment, which really just consists of us observing our media tubes and seeing if we've noticed any difference or changes in growth or movement or turbidity or anything like that. So I actually left mine 3 days. So today is Saturday, August 1st, and I did leave mine 3 days just because I didn't see really much of a difference. So this pretty much concludes the experiment portion. So I did leave these in the closet, and I think according to feedback that I received on prior experiments, the conditions were right. So hopefully everything comes out okay. So I think we're gonna end this now, and up next we're gonna do our results video, and we'll talk a little bit about how everything went and what we observed. Bye.